Perfect. And now we're moving into our third and final section of our third and final day of the workshop. We'll have two presentations for the remainder of the day. And first, I would like to introduce Dr. Jennifer Mamrosh from the Los Alamos National Laboratory to present the HIV LANL databases. Thank you so much, Jennifer.
Okay, thank you, Nina. So I'm happy to be here today. Let me just start sharing my slides. Okay, so I'm here from Los Alamos National Lab's HIV databases group, where I work with a really great group of several other individuals to produce what we call our HIV databases that have HIV immunology information and also HIV sequences in them. And we are funded by the NIAID. So HIV is a very serious health concern still. This is human immunodeficiency virus. An infection results in loss of CD4 positive T cells. And this then results in severe immunodeficiency, and individuals are very susceptible to opportunistic infections and certain cancers, and they will ultimately die of a condition called AIDS. So in the past, before treatments existed, individuals, even young healthy individuals, typically died within 8 to 10 years after infection from AIDS. And fortunately, these days with treatments, individuals can live near normal lifespans. However, it's still a serious health concern. There's no cure for HIV. About 1% of adults worldwide are HIV positive. It's quite prevalent in certain countries. It's over 20% prevalence in a lot of Southern Africa. And about 1/4 of people living with HIV are not able to be treated. So that means that they are going to die of AIDS within about 10 years unless they're able to access treatment. And there's been a huge push to develop a vaccine for HIV. This could eradicate HIV worldwide, but unfortunately, these have proved extraordinarily difficult to develop. So if you look at most approved vaccines, typically the immunogen used in the vaccine is very minimally engineered. Sometimes you can just put in live attenuated virus into a person, they'll mount a great immune response. Vaccines have been successfully administered for over 100 years. And often, for most approved vaccines, you only need one or a couple administrations of the same immunogen. And of course, these strategies were tried early on in HIV, but they were almost entirely unsuccessful. So the current HIV vaccine development strategies are now very complex. The immunogen is typically highly engineered, is engineered so that it hopefully will engage rare B cell populations. And it's expected now that you most likely are going to have to give multiple administrations, probably several different immunogens over the course of over the course of a few years to try to help shepherd antibodies to acquire improbable mutations. So the antibodies that are going to be successful against HIV are not ones that can be elicited quickly in response to a vaccine. You really, by doing a vaccination strategy, are trying to emulate the coevolution of antibodies and HIV in a host as you would have a natural infection. And when you read this, it really looks quite depressing. There has been some, I think, notable progress in the last couple of years. And this challenge is largely due to the high mutational rate of HIV. It mutates many times faster than host DNA. It mutates about 2,000 times faster than SARS-CoV-2 and about 200 times faster than influenza, which is already considered a rapidly mutating virus. And there are some additional challenges like heavy glycosylation of the HIV envelope protein and also latent HIV reservoirs. So I think together, all of this indicates that immune escape is far more pronounced and possible in HIV versus nearly all diseases. I can't even think of another disease that comes close to HIV in terms of how quickly and how readily it evolves to evade the immune system. And if you look at immune responses to HIV, it's not that they're unsuccessful. They actually are mounted pretty quickly and robustly, and they're successful at it first. It's just that the rapid immunogenesis rate of HIV eventually will render the immune response no longer successful. So if you look at this graph here, in green, this shows viral load in an individual. And viral load peaks quickly after infection, but then it is actually brought down to a lower but still detectable level. And this is due to a very rapid cytotoxic T lymphocyte response, and then a slower but very sustained antibody response shown here in blue. And these responses are able to control HIV progression somewhat in a host. They're initially actually fairly successful, but long term they are not. And without treatment, nearly all individuals will develop AIDS. And there also are CD4 positive T cell responses to HIV as well, but I think they're less well understood. They're not shown on this graph here. And only about 1% of individuals appear to be able to control HIV progression through T cell responses. And probably a slightly higher percentage of individuals make antibodies that have enough breadth to control HIV. But still, this is a very small percentage of individuals that are able to become infected with HIV and not become severely ill. So if you look at T cell responses to HIV, these, as I mentioned, are often quite effective early on. So these graphs here show two different individuals, and we're looking at just a T cell epitope sequence in these tables. And in dark blue, this is what we could consider maybe like the wild type sequence. It doesn't have any mutations yet, and there's a very effective T cell response to this epitope. However, over time, individuals start acquiring mutations in the sequence that renders the T cell response no longer effective. And you don't get exactly the same pattern of evolution between individuals, but there's a very consistent pattern of T cell responses just no longer being effective over time. And as you can see by these graphs, this is occurring over the course of several years. And I would say that, this is a well-studied T cell epitope here, but this process of immune escape is most likely occurring for many epitopes at once in response to HIV. And when you really think about it, HIV evolves within hosts similar to how cancer evolves, where there's selection for and expansion of HIV clones that contain beneficial mutations, and those are those typically facilitating immune response or immune escape. And antibody responses are similarly hampered by the mutagenesis rate of HIV. So if you look at antibody responses to HIV in most individuals, they do exist, but they typically do not have sufficient breadth to bind and neutralize evolving strains of HIV, even though they might be good binders and neutralizers of the initial strain of HIV the individual was infected with. And these broadly neutralizing antibodies do develop in a small percentage of individuals with HIV, but it typically takes about 2 to 3 years. I mean, again, this is due to like the necessity for coevolution of host antibodies along with HIV, such that the antibodies have been exposed to so many different strains of HIV that they're able to neutralize a much wider range of new strains that might develop. So our database, the LANL's HIV sequence and immunology database, have existed since shortly after the discovery of HIV. If you're interested in HIV immune evasion, I would definitely suggest going to our HIV immunology database. This one has operated since 1994, and it contains information on immune responses to HIV. And our database, along with LANL's HIV sequence database, together represent the first NIH-funded pathogen databases. So if you would like to go to them, you could go to hiv.lanl.gov. And here is just kind of an overview of how our databases are structured. And I made it very HIV immunology database centric because that's what we'll be talking about today. So this database contains information on over 4,000 HIV antibodies. Only some of them have defined epitopes, and that's, I think, a big distinction from how IEDB organizes their data and collects their data. We also have information on, I think, a little over 5,000 unique T cell epitopes. And most of the epitopes we curate are immunogenic, but we also have what we term epitope variants. And some of these could be immunogenic, some of them are not. Sometimes this is, not well characterized. And we often have information on the individual from which these immune responses were characterized from. And the HIV sequence database is at least equally important. It's highly used by the HIV research community. It has over 1 million HIV sequences now. And again, also often information on the sequence donor individual, which we call patient in our database for lack of a better accepted term. And there's some overlap of individuals between the two databases. So the immunology database is connected to a few other databases. We have what we call our antibody contacts and features database. So this has information on where antibodies bind the HIV envelope protein, because that's the main target, or other amino acids of the HIV envelope protein, and which antibodies have special relationships with. And then we also have our neutralization database that contains data on how antibodies neutralize HIV. And there's also a tool called CatNap to analyze neutralization data. And I'll give a demo of this in some of the later slides. And then we also have a large number of data analysis tools. We have tools to design vaccines, align sequences, visualize 3D structures, identify neutralizing antibodies. Many of our tools are used by researchers that are not interested in HIV. So we have a lot of just very general usage ones. I would encourage you to check these out if you haven't looked at our database before. So today's tutorials will just show through two research examples, which hopefully will be live demonstrations, how to use LANL's HIV immunology database. And I won't be able to cover everything today. These are two kind of cherry-picked examples that are pretty simple and had nice conclusions. But we have a lot of additional tutorials on our database website. Some of these tutorials are much more in depth, or they'll go through the search interfaces we have on our database website, telling you exactly what every single field is, how to enter data. Like, they are much more comprehensive in that sense. So if you are a beginner user, you may actually want to look at those as well, because I won't have time to cover all of that today. And ultimately, we are an NIH funded resource. We are funded to help you. Please email us at any time for additional help. We could set up training sessions, help you with data analysis, anything of that nature. So you could email us at immuno@lanl.gov. So for the first research example, I picked one that deals with T cell epitopes, specifically cytotoxic T lymphocyte epitopes. So in this example, they looked at individuals that could be called HIV long-term non-progressors, and it came from this paper here. So they looked at 19 of these individuals. They were considered long-term non-progressors, and this means that they had HIV for a substantially long time, and their disease was not progressing despite not being treated. All of these individuals carried the protective MEC class 1 allele HLA-B27. The individuals had their HIV gag protein sequenced at the study start and last follow-up. On average, it was about 14 years between study start and last follow-up. And unfortunately, during the course of the study, some of the individuals had their disease progress. So the researchers performing this study were interested in escape mutations, and they were looking at the HIV gag protein, and I think they sequenced the entire gag protein, and this just shows a short portion of it here. And they were wondering, do any mutations in the gag protein contribute to disease progression in certain individuals over time? Because during the course of the study, select individuals started to have their HIV progress despite having been long-term non-progressors for a long period of time. So just by looking at a sequence, you can't just pick a residue here and say, this is probably why immune escape is happening, or you're not able to immediately understand what the consequence of these mutations might be. So I'm going to show through this example how you could use LANL's HIV immunology database to investigate questions like this. So for the purpose of this talk, just to make it simple, we're just going to pick one mutation site, which is GAG amino acid R264, shown here. And we're particularly interested when it mutates to a lysine. And you could ask, how do we know which antibody or T cell epitopes might contain this mutation? And how my slides are set up now is I have screenshots just in case we're not able to do the live demos, but I think we'll be able to do them. So that's why I'll be going through the slides quickly, but I want to show you actually a live demonstration of how to do this. So what I would suggest is if you had a sequence and you had a mutation in it and you wanted to know what epitopes might contain this, especially if you don't know if it's a T cell epitope or an antibody epitope, I would suggest going to our HIV genome browser first because I think this is the most intuitive to use. So if you go to hiv.lanl.gov, you can click on immunology database, and then under tools, it is here, HIV genome browser. So we're going to pick the GAG protein. And then when you get here, you have to select what tracks. So let's pick CTL epitopes, T helper epitopes, and antibody epitopes. So we were interested in amino acid 264. We can zoom in a little bit to see the epitopes better. Can move this over a little bit more. So in orange are the CTL epitopes, so the CD8 positive T cell epitopes. And if you move this down, you can see tons of epitopes, reported epitopes from information in scientific literature span this region. So there's probably a pretty good chance that this is a CTL epitope. If you go down, the T helper epitopes, the CD4 positive T cell epitopes, some of those also could potentially contain the mutation. And then if you go down further, it's most likely not in an antibody epitope. So if you had to pick just one type of epitope, then it might be, it seems most likely to be a CTL epitope. So that already gave us some information as to where we could get started investigating this question. So what you could do in the genome browser is click on an epitope, and then it will bring you to our database and give you additional information. But an alternative way to find the exact epitope could be to go to another tool we have called the Epitope Location Finder tool, which is abbreviated ELF. So if you click on this, you can put your protein sequence here. And I'm going to put in the short sequence, which looks like an epitope, trying to make it simple today, but you could put in a much longer protein sequence containing a mutation. And then what you want to do is you want to pick your HLA restriction. We knew the individuals were B27 positive, so we'll select that and hit submit. So if you go down, a lot of sequences come up. And the ones with the green arrow by them are the ones that are presented by the HLA allele that you selected. So here's B27, and here is the sequence we put in, and it doesn't contain any mutations. Those would be shown in red. So, this is kind of a cheater's example. Like the sequence we put in was the epitope. It's not always going to be like that, but just for simplicity, we chose this. So if you click on it, you'll be brought to our database where you can read additional information. We actually have 41 records for the sequence. But let's go back to our slides. And instead of looking at what comes up by clicking on it, what I'll do is show you how to search for this sequence in our CTL epitope search interface. So say you now know what the epitope sequence is and you want to do a search for it. So if you go back to the immunology database, you click on CTL CD8 positive search. And there are a lot of fields you can select. We don't know, we don't have something that we need to select for all of them for this example, but we know what the epitope is. And we'll say for this example, results exactly match the query sequence, but you could pick something like fuzzy match or contain. You'll get a lot more results that way. We'll pick exactly match for this. I will say that if you have a long sequence and an epitope is somewhere within it, don't put it in the epitope field because it's going to be looking for the sequence as an epitope. If you're interested in, say 50 or 100 amino acids of an HIV protein, I would instead put the coordinates up here because that is, a more correct way to search for it than saying you're looking for like an 80 amino acid long epitope. So we could go down and select a few other things. We could select species being human, but we're going to pick restricting HLA allele instead. And that will just, limit it to humans because this is a human allele. So we'll pick HLA B27 and hit search. And again, what comes up is 42 matching records. So if you scroll down, you can see that they contain a lot of information. You have the epitope sequence, you have human and or the species and restricting HLA. The immunogen was HIV infection. Some of our results, it might instead be something like vaccination. It will tell you information on which experimental methods were used to determine immunogenicity. And then if you go down further, we have notes. And this is a note written by our human annotators who have read the publication, and they are summarizing what was found in the publication for that particular epitope. So, this is a much more efficient way to get information than going to the publication yourself and reading it. So we have one record per publication. And this is a little bit complex of a note. I won't read it here, but this note is a little bit simpler to understand. And it says this epitope was recognized by 3 out of 5 HLA-B27 positive patients. So already we know that this can be immunogenic epitope. So you could go down further, read some additional examples. Here, there aren't experimental methods listed. If you decided, hey, I only really care about granzyme B release in an ELISpot assay, you can select that and then hit search again. No results come up for this, but you can limit the number of results coming up by selecting just specific experimental methods. So again, I think that there's ample evidence in our database, especially if you went and read more of the notes, in the records. There are 42 records. You would find that there's a lot of evidence that this epitope is immunogenic. And here's a different example. It says 5 out of 7 B27 positive subjects responded to this epitope. So then you could ask the question, okay, are CTL epitopes, this particular epitope containing R264 mutations no longer immunogenic? Because this would explain immune escape in these individuals that are no longer long-term non-progressors. So some information along these lines is actually found in the CTL epitope search results. However, we have a separate search called the CTL epitope variant search that's more specifically set up to address these kinds of questions. So what we're going to do is put the sequence into the variant epitope search. So go back to immunology database and click on CTL variant search. So this looks like the CTL epitope search, but it's a little bit less complex. We'll put in the epitope sequence here. Again, we'll say exactly match, and we'll pick the restricting HLA of B27 and hit search. And here 8 matching records come up, and they show, kind of the parental epitope here, and then variants here. And we're interested in the second amino acid, and we're particularly interested in R264K mutations. So this is the one that we're really interested in, and it says it's calculated escape. And that might not be immediately intuitive. So if you scroll down, it gives you more information on what this means. And it says this variant was more often found in HLA matched than HLA unmatched patients. So this doesn't directly demonstrate immunogenicity. If you go down, you can find some other types of examples. So here's the mutated epitope that we're interested in again. If you go down, you can read some more information. And here they actually did interferon gamma ELISpot assays. And it says here that no response to this epitope was seen in B27 positive individuals. So I think that this really demonstrates that the epitope by itself that's not mutated is immunogenic, but it seems that R264 mutations are fairly common, and they also can confer immune escape in individuals with this HLA. So hopefully this gives you a sense as to how you could use our database. Like say you had the sequence of an HIV protein and it's like 400 amino acids long, you have no idea what kind of epitopes might exist within it. Hopefully this gives you a sense as to how you can actually find specific epitopes that might be mutated and might become escape mutations, even if they're within a really long sequence. And I won't go through this here, but if you want a quick summary of how to search for CTL epitopes or T helper epitopes in our database, if you look at the YouTube slides when they're released by IEDB in a couple of weeks, this will hopefully be helpful to you as a resource. So then let's move on to the second research example. So this one involves antibody responses to HIV. So as I mentioned before, in response to HIV infection, a small percentage of individuals will develop, and this takes a couple of years, what are called broadly neutralizing antibodies. And these can be quite potent. These have been identified from individuals. Some of them are given therapeutically to individuals, and they're able to control HIV infection when given. However, they don't usually work forever. I mean, they may work for a couple of months, and then that individual's HLA, HIV in their body will mutate away to evade being targeted by the broadly neutralizing antibody. And something that I want to point out before we move on is that, of course, antibody epitopes are often discontinuous, and for HIV, they're typically against specific regions of the envelope protein. And some of these regions are shown here. And our database actually contains a pretty small number of exact epitopes. We only know the exact epitope for about 10% of our antibodies. Some of our antibodies, we don't know really anything about the epitope. For a lot more of them, we might know that they're a CD4 binding site antibody. We know the amino acids that constitute the CD4 binding site, but we don't actually know whether the antibody binds all of those amino acids. It most likely doesn't, but it will bind some of them. So unlike IEDB, we often don't have a specific epitope for some of our antibodies. So if you go to search for an epitope in our database and nothing comes up, but you know the epitope is somewhere within the V3 glycan region, you could instead search for V3 glycan antibodies. And that might give you some that could actually bind the epitope, even though it wasn't reported, a specific epitope wasn't reported in the publication. So for this research example, which came from this paper here, it is actually a really cool study. The researchers took libraries of HIV containing single point mutations in the envelope protein, and they incubated them with one type of broadly neutralizing antibody at a time, and they gave HIV some time to mutate away from antibody selective pressure. And then what they did is they characterized which mutations seem to allow for viral escape from the antibody. And the researchers looked at several different broadly neutralizing antibodies. We're just going to look at one today called 10-1074. And they characterized ONV amino acids that seem to be mutating in response to antibody selective pressure, in the sense that they're conferring immune escape. And there are really three main sites that mutated. It was D325, N332, and S334. And you can see the amino acid that it mutates to, it doesn't really matter. I mean, it's not something that seems like a gain of function type of mutation allowing for something new to happen. It's most likely that you're losing the amino acid that the antibody can bind to, and it doesn't really matter what it's replaced with, but once it's replaced, then the antibody no longer is able to bind, most likely. But we don't know this for sure. I mean, that's just, a reasonable speculation. So the question that this example will try to ask is, can our immunology database give us any insight into why these sites might have mutated? So what we could start with, because we actually know the antibody of interest, is just doing an antibody search in our database for this antibody. So we can go back to the immunology database and let's click on antibody search. And a lot of this information we don't know, but we know we're interested in the 10-1074 antibody. We'll click exact match, and then we'll just hit search. And one record comes up. It is indeed the antibody we're interested in. And instead of how the T cell epitopes have one record for each publication, we have one record for each antibody. And we actually have information from 88 publications in this record. So if you look at what comes up, there's some, fairly simple information on what protein the antibody was raised against. We know it's a monoclonal antibody. It binds to this region of the envelope protein. It's neutralizing. It's a human antibody. HIV infection was the immunogen. If you click here, there's information on the antibody donor. And if you go down to the notes section, each of the notes comes from a separate publication. It's notes, they're not just copy and pasted from the paper, they're notes that were written by our human annotators who have read the whole paper very carefully, and they've summarized the key findings in just a couple of sentences. So again, this is, I think, a very efficient way to get information quickly for any new antibody that you're interested in. But we're really interested in specific regions of the HIV envelope protein. So I would suggest clicking on View Contacts and Features to get information about this for this antibody. So our contact and feature database came up here, and there's different types of features listed here. We're going to be interested in contacts because that's really where the antibody is binding the envelope protein. And you can see already that some of the sites that we care about, like 325, 332, and 334, are listed here. So, these are antibody contact sites. That makes it less surprising why these sites might have mutated in response to antibody selective pressure. And if you click on that link, you'll get more information as to like what kind of interaction it is. 332 here, the amino acid 332 says it's a primary glycan contact. So the antibody is actually interacting with the glycan. And there's information for additional publications below. So what we can find, from just this quick and initial search is that our database has a lot of information for this well-studied antibody. This N332 site is a glycosylation site, and this antibody is known to contact some of the sites that we're interested in for this research example. So I would say, it really isn't surprising that ONV has mutated at these sites in response to antibody selective pressure. So you could ask questions like, do other antibodies target these residues? So if you go back to the contacts and features database, we could do a new search by clicking on feature database here. You could select an antibody here. We don't want to just pick 101074 again, but we could instead here type in amino acids that we're interested in. We'll type in 325 and 332. For feature type, we'll pick contacts and then we'll hit submit. And at the top is 101074. So this antibody is known to contact these sites, as we just found, but there are several other antibodies that contact them as well. So you would expect that if an individual was being treated with 101074 and they became resistant to it, you probably wouldn't want to give them these other antibodies therapeutically because there's a pretty good chance that they would be resistant to these as well because they contact similar sites of the envelope protein. So if you wanted to find antibodies that would be likely to work in an individual that develops a resistance to 101074, you could go back to the contacts and features database and you could maybe select different types of antibodies that don't target the V3 glycan region, and see if those would be more useful to use in these patients. But we actually have a better way of doing this. And a better way to determine this, like say you wanted to find an alternative antibody to treat somebody with, would be to use our CatNap tool. And this ultimately is aligning viral sequences with antibody neutralization assay values. And you could use this tool to find antibodies with neutralizing activity, even against viruses that have, say, D325, N332, or S334 mutations. And I don't have time to cover it today, but CatNap has a lot of additional useful features. You can actually do significant analyses of specific amino acid positions to see if a specific amino acid is associated with neutralization activity. We have information on antibody sequences and contact sites, but I'll give you a quick demonstration of CatNap right now. So if you click on databases, go to neutralization database, let's click on CatNap. And this is a complicated tool because it has tons of data in it. We have tens of thousands of neutralization assay data points in it. So I will try to keep this example as simple as possible. So let's click on number of viruses tested. And on the left panel here are antibodies. And I'm just going to pick the 10 most commonly tested antibodies. And this includes 1074. So I've checked just the top 10, most frequently studied ones. And then this rightmost panel is viruses. And we have over 2,500 viruses that have been assayed. We don't want to look at all of them just because it's going to be overwhelming for such a short demonstration. So we'll click on panels and we'll go down to this virus panel that's called the global panel. It's supposed to be representative of HIV strains worldwide, and there are only 12 viruses in it. And we'll go down and hit submit. And what comes up looks kind of complicated. I mean, it actually could look much more complicated if you selected more viruses and antibodies. So on the left here are the virus names. In the middle panel is each antibody, the neutralization assay value. So here's 1074, and we can sort the columns by how well the antibody neutralizes. So these values are quite low, which means that this antibody neutralized all of these viruses very well. However, it had almost no activity or no activity against a couple of the viruses. And you can look at the other antibodies that we selected. Some of them actually, like this one, it isn't like 1074. Like it has very good neutralizing activity even against these viruses for whatever reason. And this antibody, PGT121, is very much like 1074 for reasons, we don't immediately know. So say you're wondering, okay, like why are these viruses able to be neutralized and these ones are not? Does it have something to do with specific amino acids in the virus? So on the rightmost panel shows alignments of the virus sequences for the envelope protein. So we can scroll over, and I'm going to pick the most simple example possible. We're going to look at the N332 amino acid. And it shows, the N shows up in red when it's a predicted glycosylation site. And you can see here that the presence of a glycosylation site at 332 is very highly correlated with neutralization activity of this antibody, because these viruses down here, these 4 viruses, don't have a predicted glycosylation site, and this antibody has, virtually no activity against them. So this really, I think, gives you a sense as to why this antibody could be effective against these strains of viruses, but not against these. And if this antibody no longer worked in an individual, you could pick a different one. I mean, if you look at this antibody here that has very good neutralization activity against pretty much all of these viruses, this one is very agnostic to whether a glycosylation site is at this amino acid or not. It most likely might somewhere else on the envelope protein. So an antibody like this would be a good choice to give therapeutically. So again, here's a resource you can go back to in the future if you're looking at the slides on YouTube. And I just wanted to say our databases contain dozens of additional data analysis tools again. We have vaccine design tools. These are used by a lot of researchers that are not HIV researchers. We get tons of questions from users about using these for other viruses, which you absolutely can. You put in protein sequences and we'll generate synthetic vaccine cocktails that importantly try to maximize the epitopes chosen based on the population frequencies. And it's not giving you consensus epitopes, it's giving you actually occurring epitopes. We have an HIV 3D viewer, which is probably not the best name for it because you can actually upload your own PDB files in it as well, but it has a lot of additional features for viewing HIV proteins, where it has some boxes you can check to color specific regions of the envelope protein, for example. We have a lot of tools useful for HLA analyses. tools to calculate HLA frequencies, say comparing different populations. You can find HLA allele motifs and sequences. You can identify epitopes in a sequence based on the HLA allele frequencies found in a population. And we also have a similar CatNap tool for COVID-19. We have this tool and a few other things related to COVID-19 available at cov.lanl.gov. So if you're interested in COVID-19, please check this out. And lastly, most of our data is going to soon be available at IEDB. So we've started to convert some of our records to IEDB's format. Some can be easily converted, like epitope sequence. Some records, like information in our notes that's written by a human, that's really difficult to convert in an automated manner. So we will have our data converted to IEDB's format as best as possible so it can be retrievable through IEDB searches. But all of IEDB's records will contain a link back to our database. And this will give you our complete record, including things like our handwritten notes. And it also will give you access to HIV-specific tools or capabilities. For example, CatNap. You wouldn't want to download tens of thousands of neutralization assay values and analyze them in Excel. It would be much more efficient to use our CatNap tool. So thank you for attending. We are funded to help you. Please at any time contact us with training requests, questions, problems, or suggestions. Immuno@lanl.gov. Thank you.